Hello guys, good morning. Our topic is mass spectrometry and determination of 3D structure of protein, X-ray crystallography, X-ray diffraction. Myself, Jumana Karadan, second MSc Biology from St. Mary's College, Thrissur, in the guide of Dr. Kain Vadakan, Assistant Professor, Department of Biology. Mass spectrometry. Mass spectrometry is an analytical technique that separates ionized particles such as atoms, molecules, and clusters by using difference in ratio of their charges to their respective masses and can be used to determine the molecular weight of the particles. Working, a mass spectrometer can measure the mass of a molecule only after it converts the molecule to a gas phase ions. To do so, it imparts an electrical charge to the molecules and converts the resultant flux of electrically charged ions to a proportional electrical current that a system then reads. Parts. A mass spectrometer comprises three parts: ion source, a mass analyzer, and a detector. The sample to be analyzed is ionized in the ion source. Ions are then transported by magnetic or electrical field to the mass analyzer. Yeah, here you can see the sample injection port, and the sample molecules get vaporized in the presence of the electron beam, and they were accelerated through the slit and then transported through the magnetic field which is created by the magnet and then detected by the detector. Stages. The four stages of mass spectrometry are ionization, acceleration, deflection, and detection. The sample is vaporized before being passed to an analyzation chamber where it is bombarded by a stream of electrons emitted by an electrically heated metal coil. Advantages. Mass spectrometer has three major advantages: the capacity to study numerous molecules regardless of whether they are from the same structural family or not, the capacity to highlight the specific metabolites of a disease. It's an automated technique offering the possibility of large scale analysis. Now we have to study about the determination of 3D structure of protein. 3D dimensional structure of a protein is determined by techniques such as X-ray crystallography and nuclear magnetic resonance. Knowledge of protein's 3D structure is a huge hint for understanding how the protein works and use that information for different purposes: control or modify protein functions, predict what molecule bind to that protein, and understand various biological interactions, assist drug discovery. X-ray crystallography. X-ray crystallography is the experimental science determining the atomic and molecular structure of a crystal, in which the crystalline structure causes a beam of incident X-rays to diffract into many specific directions. Yeah, here in the structure, here in the figure you can see a purified protein which is undergo crystallization, and crystals of proteins formed and undergo the process of X-ray diffraction, and we get the electron density, and that is where the 3D structure of protein is obtained. X-ray crystallography uses X-ray diffraction method, a scattering of X-rays by the atoms of crystal that produces an interference effect so that the diffraction pattern gives information on the structure of the crystal or the identifier of a crystalline substance. Here you can see incident X-rays and diffraction X-rays on a particle. Bragg's law. Bragg's law states the following: when X-ray is incident onto a crystal surface, its angle of incidence, that is theta, will reflect with the same angle of diffraction, that is theta. And when the path difference is d is equal to the whole number n of the wavelength lambda, constrictive interference will occur. Instrumentation. In a single crystal X-ray diffraction measurement, a crystal is mounted on a goniometer. The goniometer is used to position the crystal at selected orientation. The crystal is illuminated with a finely focused monochromatic beam of X-rays, producing a diffraction pattern of regularly spaced spots known as deflection. Here in this picture, you can see X-ray source, and this undergo through the slit, that is the monochromator, which means a single wavelength of light can be only passes when it passes through the monochromator. Yeah, then which enter to the sample and undergo diffraction of the X-rays, which is detected on the refraction detector, and we get the absorption detector. Application. X-ray crystallography can reveal the precise three-dimensional positions of most atoms in a protein molecule because X-rays and covalent bond have a similar wavelength and therefore currently provide the best visualization of a protein structure. Thank you.